Thanks a lot. So really excited to talk about what we do in our lab. And so the title is Evaluating the Robustness and Reproducibility of RNA-Seq Quantification Tools. So small disclosure, so the work done by undergraduates who was admitted to USC. And so it should be a very simple project. It's a kind of undergraduate project, so we'll keep it simple. So when we talk about robustness and reproducibility, I think genomics is an interesting field because, and I think it's quite different from computer science, because when we talk about result reproducibility, and this is what you can see right here, right? Meaning that, like given the same input, running the tools, can you achieve the same results, right? But the input is the same. However, in genomics, I think it's quite different, because if you think about genomic-based medicine, or whatever we call it, personalized medicine, right, we expect some experimental variation, right? Let's say we go to the doctor, right, and we want to get the same kind of a report, and we want that the genomic results to be used in the same way to prescribe the drugs or decide on the therapy. And then what that requires, we want the results from sequencing, whatever the other experiment, to be the same if we do it here in Lyon, and let's say we do it all the way in Los Angeles. However, we know that's not the case, right? So we know that there is some experimental variation in the sequencing data, right? Even the sample is the same. And the question is, can we achieve the same results, right? The same bioinformatics results. And I think this is what we mean here. We try to kind of visualize it. This is what we call, oops, sorry. Yeah, so like it's here. So this is what we call genomic reproducibility. We didn't find a better term, right? So we decided to invent one. So then given the differences in the data, right, can we achieve the same bioinformatic results? So we were curious to apply that to RNA sequencing and gene expression quantification. And the way to do that, there are kind of fundamentally two ways to do it. One is to generate the real replicates, right? Meaning you take a sample and you sequence in one laboratory and another one and so on. But as we know, that's expensive, right? And then it's time consuming. And sometimes it's impossible if it's some clinical restricted data in the clinical settings. So what we're suggesting some proxy of the real replicates, what like technical replicates or computational replicates, I guess better say. So those are alternative to the real replicates, and they're computationally generated. And the downside of that, as we know with simulation data, it's can never capture the true kind of variation in experimental data. So it captures some part of experimental variability. So the way we do that, and remember that's undergraduate project, right? So we do it in the very, very simple way. How about we just resort the reads, right? So how about we require bioinformatics tools to maintain the same results if the reads are resorted? I think fair enough. How about reverse complement? We take every read and we make a reverse complement, and we expect the bioinformatics tools to give the same results. So those two types, right? We have type number one, which is resorting. Type number two, we take reverse complement with the read. So what we did, we took a couple of like 6 so far paired end RNA-Seq data from 1000 Genome Project of 75 base pairs length, paired end data, and we will consider many transcriptomic quantification tools. So far, we have got Selman and Callista, which are both pseudo-alignment tools. And we ask the question, will they maintain the same gene expression estimates across those technical replicate generated computationally? Right, so we did 3 times. We can do perhaps more. So we did 3 times shuffled reads, so change the order of the reads, and we took once reverse complement. And this is what we see so far, right? So many genes have inconsistent gene expression across replicate number 1, right? Number 1 when we resort. And it's quite similar, right? You can see Selman and Callista. I mean, there are some differences, but there are many genes, right? Actually, the results are a little bit different if you talk about reverse complement. So you can see Callista perhaps is doing a better job than the Selman, right? So there is some differences. So if you want formally kind of look at that, so I suggest to first look at the exact match. How about we require precisely the same gene expression, right? You can see that around 45% of the genes will have identical gene expression, but the rest will be either within 1% difference, right? So the difference between original and then original data and the replicates will be 1%, or it's actually interesting that quite many genes are beyond 10%, right? you can see that there are 30 or 45% of the genes which are more than 10% difference between the original and replicate, quite a bit. So we kind of think how is that can be, because it's quite high number. So we investigate. So we hope we didn't make any kind of mistakes and so on. So we were quite surprised, right? We're quite surprised why is that happening. Then we look at the 100 mostly affected genes, and we want to know, maybe there is something specific about those genes. Maybe just they are so hard. So what we see, there is quite substantial number of genes which are affected, right? Which are the most, where we observe the most discrepancy in the expression for the reverse complement and for the shuffled version, right? And what are those genes? You can see the names, or at least the gene IDs, and you can see they are not consistent, right? So for the Callista, so you take a sample, right, and you see that the most affected gene is different, right? And sometimes it's the same, right? For the last sample, it was the same gene being affected, but very different behavior for the salmon. So there is one particular gene salmon doesn't like, right? So somehow it's always affected, right? Like, and then you can see it almost goes from 0 to some value, right, in expression level. So somehow there is no reads being assigned through the k-mers, right, to that gene originally, but somehow after you sort the reads, something happens, right? So we're trying to investigate and get into the algorithm, try to understand what's going on. So this is very, very kind of preliminary work. And I think just to be on time, so I think so far what we see, so as you can see, there is substantial variability in the ability of RNA-Seq methods to maintain consistent gene expression across computational replicates. And we hope that will be the study we do will be kind of useful and important, because to me, we talk a lot about accuracy, right? Like when we write the method paper, when we develop the method and we publish the method, we really, for the good, right? Like we really focus on the precision, sensitivity, accuracy, F-score, and so on, which is great. But we kind of ignore what that we call genomic reproducibility, and we kind of recognize the challenges of generating the data, right? Like not every one of us is capable to generate it, and as we mentioned, sometimes it's impossible. But hope we can provide like framework which can do some framework which we can assess genomic reproducibility for computational replicates. And I think that's the line I was putting to all my talks. So our research is dedicated to freedom-loving people, and I did it and people of Ukraine who fight for freedom. So I did it last year, I do this year. Hopefully I will not do that line next year. Okay, so, and then thanks for all the students in the lab. So, and then we're hiring for the postdocs. So if you want to ski and surf in the same day, I think California is for you. So come join our lab. Thank you.
Thank you. We have time for one question. Okay, there's one. There will be chance to chat with Serghei also after, but we need to be
I'm coming
strict
to
on
the
time.
dinner. So if you guys are around, happy to.
So I just don't know if Callisto and Salmon, I'm over here.
Oh yeah, okay.
Do they, if you were to repeat exactly the same input FASTQ files, do they give various results? Is there like a level?
Yes, so we did that, and yes, sorry, I didn't put the slides. And at least for the data for those 6 samples we did, and the results were the same.
Okay.
Consistent. So if you don't alter the input, the results stays the same.
Okay, so it's definitely the shuffling and the reverse.
Right. So this like seems like that's exactly the shuffling.
Okay. Just,
So Callisto has a seed option because it's an optimization algorithm. And did you try to change the seed of the algorithm to see if because you can do several inference replicates using Callisto?
Yeah, that's a great question. And that's something we would like to do. So we'd like to explore the parameters and see if there is a specific parameters which are causing this, like including the seed length. Right, like these specific parameters influencing the result.
It's a random seed. It's because it's a random algorithm. So maybe it's the way the order of reads. It just affect the random number generation and gives different results.
Right. And is it random also for Salmon? I guess so, right?
Yeah. Yes, because it's likelihood optimization.
Right. Okay. Yeah.